Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, everyone. My name is Tsabita. My student ID number is 13086 from Biology A 2020. In this presentation, I want to talking about viruses. Definition: what is a virus? Viruses are submicroscopic, which means that you cannot see them in a microscope. What's interesting about viruses is that they have two or three components. Starting from the inside, you will have a nucleic acid, which can be either DNA or RNA, and in both cases, the nucleic acid can be either single-stranded or double-stranded. Then surrounding the nucleic acid will be a protein coat that's in the form of capsid, or little small units that are assembled in certain way. That's what all viruses have. Now, some viruses will also have an envelope which they obtain as they emerge from the cell. Viruses are very interesting in that they can only survive inside a living cell, so they must have a living cell in order to survive and replicate. The next is the structure of viruses. There are a lot of different viruses in the world, so viruses vary in their size, shapes, and life cycle. Viruses do, however, have a few key features in common. These include a protective protein shell or capsid, a nucleic acid genome made of DNA or RNA cooked inside of capsid, a layer of membrane called the envelope. Some, but not all viruses have. Let's take a closer look at this feature. This is a nucleic acid genome. This is the capsid, and this is the envelope. The next is viruses capsids. Here, there are three kinds of virus capsids. The first is icosahedral. Icosahedral capsids have 20 faces and are named after the 20-sided shape called an icosahedron. The second is filamentous. Filamentous capsids are named after their linear thin thread-like appearance. They may also be called rod-shaped or helical. The third is heptile. These capsids are kind of a hybrid between the filamentous and icosahedral shapes. They basically consist of an icosahedral head attached to a filamentous tail. The next is virus envelopes. In addition to the capsid, some viruses also have an external lipid membrane known as an envelope, which surrounds the entire capsid. Viruses with envelopes don't provide instruction for the envelope lipids. Instead, they borrow parts from the host membranes on their way out of the cell. Envelopes do, however, contain proteins that are specified by the virus, which often help viral particles bind to host cells, which often help particles. Although envelopes are common, especially among animal viruses, they are not found in every virus. Okay, the next is what's a viral infection? In everyday life, we tend to think of a viral infection as a nasty collection of symptoms we get when catch a virus, such as the flu or the chickenpox. But what's actually happening in your body when you have a virus? At the microscopic scale, a viral infection means that many viruses are using your cells to make more copies of themselves. The viral life cycle is the set of steps in which a virus recognizes and enters a host cell and reprograms the host by providing instructions in the form of viral DNA or RNA and uses the host's resources to make more viruses particles. The output of the viral program for a typical virus, the life cycle can be divided into 5 steps. The details of these steps will be different for each virus. The diagram shows how these steps might occur for a virus with a single stranded RNA genome. This is a general diagram of a virus life cycle. The first is attachment. The virus recognizes and binds to a host cell via a receptor molecule on the cell surface. The second is entry. The virus or its genetic material enters the cell. The third is replication and gene expression. The viral genome is copied and its genes are expressed to make viral proteins. Is the fourth is assembly. The new viral particles are assembled from the genome copies and viral proteins, and the last is release. Completed viral particles exit the cell and can infect other cells. This is to infect new cells. Okay, the next is virus genome. All viruses have genetic material or genome made of nucleic acid. You, like all other cell-based life, use DNA as your genetic material. Viruses, on the other hand, may use either RNA or DNA, both of which are types of nucleic acid. We often think of DNA as double-stranded and RNA as single-stranded, since that's typically the case in our own cells. However, viruses can have a possible combos of strandedness and nucleic acid type, double-stranded DNA, double-stranded RNA, single-stranded DNA, or single-stranded RNA. Viral genomes also come in various shapes, size and varieties. Do they are generally much smaller than the genomes of cellular organisms. Notably, DNA or an RNA viruses always use the same genetic code as living cells. If they didn't, they would have no way to reprogram their host cells. Okay, that's all. Thank you for your attention. If I have many mistakes, forgive me if I have many mistakes. The last I say, wassalamualaikum warahmatullahi wabarakatuh.